This video is going to recap DNA and DNA replication. We'll look at organisation of DNA in eukaryotes and prokaryotes. Then we're going to look at the structure of DNA and how DNA is replicated prior to cell division. So we're going to start by looking at how DNA is organised inside cells. We learn in Nat 5 about the structure of DNA as a double helix, but there's actually lots of different ways that DNA can be organised in a cell depending on what type of cell it is. And the first one that we're going to talk about are prokaryotes, and these are things like bacteria. And they have two ways that they organise their DNA. They have a single circular chromosome, so this is this bit here with the sort of squiggly lines, that is your circular chromosome. And they also have these smaller circular plasmids, so you remember talking about plasmids when you did genetic engineering last year as well. They don't have a nucleus, so we don't find linear chromosomes like we do in a DNA double helix. So eukaryotes, on the other hand, are things like plants, animals, and fungi, and they all have linear chromosomes that they find within their nucleus. So think of your DNA double helix, that would be found in a linear chromosome. And they are really tightly coiled and they're packaged with these associated proteins called histones that we're going to look at in a little minute. They also have these circular chromosomes, so we can see that this mitochondria has been magnified here, and we can see that it has those little squiggly lines again. Those are your circular chromosomes. And in plants, we would find those circular chromosomes within their chloroplasts as well. Yeast is a special example because, like prokaryotes, they have plasmids. So they're your kind of special example of eukaryotes. So a common question about where DNA can be found in eukaryotes, it could be the nucleus where you'd find your linear chromosomes, mitochondria and chloroplasts you'd find your circular chromosomes, and then plasmids in yeast. And this is what I was referring to a second ago. I said that the DNA was really tightly coiled and it was packaged with these associated proteins. So we call these histones here. Okay, so moving on to talking about the process of DNA replication. Again, in Nat 5, we talk about DNA replication really briefly in the mitosis topic. And this process is occurring prior to cell division, and it's where we replicate the entirety of the genetic material so that each of these cells that we are producing during mitosis are genetically identical. So really important because it's ensuring that an exact copy of the genetic material is passed on to the daughter cells during mitosis, and that is going to ensure that the cell functions correctly. Okay, so starting to look at the structure of DNA. DNA is made up of DNA nucleotides. So what we're looking at here is a single DNA nucleotide. And it is made up of three components. So we have a base, we have a deoxyribose sugar, and a phosphate. So same as what we learn in Nat 5, the base can be one of the four bases, so A, T, C, or G. We don't need to know the full names, so adenine, thymine, cytosine, and guanine. Attached to that we have a deoxyribose sugar, and attached to that we have a phosphate. Now, you'll have noticed that I've got some numbers written on there, and what those are representing are carbon atoms. Now, I'm going to be referring a lot to something called the 3' or the third carbon end of the DNA, and the 5' or the 5 carbon end of the DNA. Now, what I'm referring to is this bit here. So the 3' end we find on the deoxyribose sugar, and then the 5' end we find on the phosphate. And we're going to be talking about the direction of DNA replication. We always add DNA nucleotides to this 3' end, and that will make a little bit more sense in a second. So I've gone ahead and I've added another DNA nucleotide to this DNA, and we need to know how these DNA nucleotides are joined together. So we can see we've got our 5 carbon phosphate of our first DNA nucleotide, and it is joined to the 3rd carbon of the deoxyribose sugar of the next DNA nucleotide. And this bond is a strong chemical bond. This is what forms this really strong sugar phosphate backbone of our DNA. So now we need to start looking at the complementary strand of DNA. So the other strand runs in the opposite direction, and we call this anti-parallel. And we can see here that the 5' end of this strand is here, and on the other strand your 5' end is here. So that is what we mean by anti-parallel. They run in opposite directions. And the other thing to take note of is the bond that is between the bases. So we're going to want, when we start to replicate our DNA, we're going to want our DNA strands to separate. So we don't want a strong chemical bond, we want them to be weak hydrogen bonds. We want them to be able to come apart really easily. So weak hydrogen bonds in between the bases. And here you can see the other complementary nucleotide that has been added with the weak hydrogen bonds in between the bases. So for example, if this base was adenine, this would be thymine. If this was cytosine, this would be guanine. And it's this base sequence of the DNA that forms our genetic code. So here I've drawn a short section of DNA that has become unwound. So we've broken those weak hydrogen bonds in between the bases, and we now have two template strands that are running anti-parallel to each other. And the way that we replicate each of these strands is going to be different because of the direction that we add DNA nucleotides. So the first thing that we are going to need in order to start this process of DNA replication is we need something called a primer. So a primer is just a short strand of DNA nucleotides, and it acts as a start point for DNA replication. So we say that these primers always bind to the 3' end of the template DNA strand. So I've just drawn it as a little box, but we need to imagine it being made up of these nucleotides. Now, when that primer binds, what it is doing is it is acting as this start point for a little enzyme called DNA polymerase. Now this DNA polymerase is going to be adding DNA nucleotides to this 3' end of this new strand that we are forming. So it's going to add, we've got C here, it's going to add a guanine. Here we've got G, we're going to add a C. It's going to keep moving along, and it just adds these complementary nucleotides. Now this strand that we are forming here, we call this the leading strand. And it's really easy to recognise because you're going to have one primer, and we're going to be adding these nucleotides in a continuous direction. Now, it doesn't actually matter if I unwound this DNA even further and I exposed more bases. This DNA polymerase would just be able to continue to move along and add more DNA nucleotides. So again, DNA polymerase is adding DNA nucleotides to the 3' end of that new DNA strand that is forming. Okay, so now we're going to look at the other side of the DNA, and remember that they are running in an anti-parallel direction, so the way that we replicate it is going to be slightly different. We still start off with a primer. So we've got our little primer binding there, and again that's going to allow DNA polymerase to bind and to start adding those DNA nucleotides to the 3' end of the new strand that is forming. And that leaves us with a little bit of an issue because we're adding nucleotides in this direction, but we still have this little section that we still need to replicate. So in order to do that, we need to add another primer. So primer is going to come along, DNA polymerase is going to be able to add DNA nucleotides to the 3' end of the new strand that is forming, and it is going to replicate that little section, or we call that a fragment. Now what we've ended up with is two little fragments of DNA there, and we're going to talk about another enzyme that is involved. We've got an enzyme called ligase, and that is going to come along and it is going to join together those fragments. And we call this strand our lagging strand. So if we're asked why are the strands replicated in opposite directions, it's because DNA polymerase can only add DNA nucleotides to the 3' end. So the other thing that we need to know about is we need to know the 6 things that are actually required for DNA replication to occur. So we need our DNA template. So that is the thing that we are actually replicating. We need DNA nucleotides. We need our enzyme DNA polymerase that's going to add those DNA nucleotides. We need primers to act as that start point for replication. We need ligase, and we need ATP because this process requires energy. So key things to think about when we're trying to identify the different strands. With your lead-in strand, you're just going to see one primer, and we're going to be adding nucleotides continuously. Whereas with your lag-in strand, you're going to see fragments, you're going to see multiple primers, and it's going to be discontinuously. We're going to see those fragments of DNA.